happened? Good morning. Okay, we're gonna wait just another minute because some people are still joining us. Okay, we're gonna go ahead and get started. It looks like most everybody is here. So let us get the right PowerPoint. We are gonna be going over the cell and cell structures today. We are a little bit ahead of schedule, so we're gonna try to take it slow, but it's always better to be ahead than behind. Okay. Okay. Cell and cellular structures. So, oh, just this thing. Sorry, I need to move that. Okay. Oh, I'm sorry. I need to check one thing. Okay, we're good. Okay, so here is a drawing depicting an animal cell, what you would typically find within it. I know this is a physiology class, but there will be a small portion in your next quiz or exam where you will be expected to identify the major structures of the cell. So I would likely use this picture, and we are going to be talking about these structures today, but ones that I would likely quiz you on would be the Golgi apparatus, mitochondrion, the smooth endoplasmic reticulum, the rough endoplasmic reticulum. Well, yeah, you should know the plasma membrane, the nuclear envelope. We'll talk about all of this, the nucleolus. The centriole, microvilli. Back over here, nuclear pore. So some of these things, they just look like little bubbles in the cell, and it's hard to distinguish the difference between a peroxisome and a lysosome, for example, if you're identifying things. So because of that, I would never ask you to identify those because it's a bubble. It doesn't have any really defining characteristic. Oh, down here, free ribosomes. So these little dots, those represent ribosomes. So I would expect you to be able to know that. So we are going to be talking about these different cellular structures today. Pardon me. And how they function. So we're going to start with the nucleus. Okay. The nucleus is the largest organelle, and it is the cell's control center. Much like the brain is our control center for our body, the nucleus is the control center for the cell. Surrounding the nucleus is the nuclear envelope. And the nuclear envelope, here we go. Sorry, I will drink more coffee. The nuclear envelope is a double membrane that surrounds the nucleus. And in it, or in the nuclear envelope, you have nuclear pores. And this allows information to pass to and from the nucleus. The perinuclear space is the area between the two layers of the nuclear envelope. I have my coffee right now. I'm sorry. So just knowing that the perinuclear space is there. The nuclear pores, I pointed those out to you already. Here are some more. This is information passageways between the inside and outside of the nucleus. And we'll talk more about what kind of information is passing through there in a little bit. Okay, major components of the nucleus, you have the nucleoli. And this is where it transcribes or synthesizes rRNA or ribosomal RNA and assembles those ribosomal subunits. So ribosomal RNA, there's two basic subunits. When they come together, they form the ribosome, which is the machinery of where protein synthesis occurs. So in the nuclei, those subunits are formed and assembled. Then they leave the nucleus through those nuclear pores and then out in the cytoplasm or attached to rough endoplasmic reticulum, protein synthesis can occur. So rRNA exits the nucleolus and enters the nucleus. So the nucleolus, if you're looking at a picture, let's just back up really quick. The nucleolus is the dark portion inside the nucleus. And this is where you have really condensed DNA material. So RNA exits the nucleus, RNA exits the nucleolus, enters the nucleus portion, then exits the nucleus through those nuclear pores, and then in the cytoplasm, to be more correct, in the cytosol. Cytosol is the fluid inside the cell. That's the specific name for the fluid. Cytoplasm is the cytosol and the organelles that are in the cell. So if you're just talking about the fluid itself, it's cytosol. So the RNA exits nucleus, and in the cytosol, either free or fixed, it carries out protein synthesis. Okay, another component of the nucleus is chromosomes. Chromosomes are made up of DNA molecules. So here is just showing you the organizational structure of chromosomes. So if we start down here at the bottom where we just have a strand of DNA, it starts to compact and it coils around these proteins called histones. And it coils and coils and coils, and then it starts making bigger coils until you get this, what's called a supercoiled region. And that happens to the entire chromosome. You form a chromosome. And so that's just very tightly packed DNA that's packed around histones and coiled around on itself. The only time that you would see chromosomes in a cell is during cell division. The rest of the time, the DNA is more unwound, like you see here. So you only see the chromosomes during cell division. Chromosome anatomy, you have what are called sister chromatids. So each one, so that is one sister chromatid, and there is the second sister chromatid. They're held together at the center portion called the centromere. Down below here, this colorful picture is showing a karyotype. And this is just all of the DNA, the chromosomes for an individual. And the colors represent genes, specific genes and areas. Looking at this karyotype, can anybody tell me if it's a male or a female karyotype? You can put it in the chat if you don't want to speak up. Male or female? Glad you guys had a good Labor Day. Okay, so we have some people not sure. Okay, how do we tell if it's a male or a female? So right down here, the 23rd, whoops, I'm sorry, let's back up. Right down here, close that. Right down here, we have, why is my thing not drawing? There we go. We have the 23rd set of chromosomes. So this one shows an X and a Y. So XY is male. If it showed XX, it would be female. So this karyotype is for a male. So good job, guys. Okay, moving on to organelles in the cytoplasm. So a little bit more about ribosomes. We know that ribosomes, their subunits are from rRNA, ribosomal RNA. They are made in the nucleolus, and then they exit through the nucleus into the cytosol, and then they perform their functions. So ribosomes specifically synthesize proteins, and they develop from that rRNA. So you can have either free ribosomes, which are just floating in the cytoplasm, which is kind of what you see in this picture here. Those black dots, the big black dots, are ribosomes. And they are able to manufacture proteins in the cytosol directly. And we're going to be talking about protein synthesis today, and I do have a couple helpful videos to help with these illustrations, because some of it is kind of hard to imagine it without some extra help. So we got a little bit of extra help today. Okay, fixed ribosomes are attached to the endoplasmic reticulum. That's what the ER stands for. Let me see if I can write that. Endoplasmic reticulum. But we do abbreviate that as ER very often, okay? So when they're fixed, this is what it looks like. You have these folded membranes, and on the outside of those membranes are those black little dots. Those black little dots are the ribosomes. So in this example, it's rough endoplasmic reticulum because it literally looks rough with those ribosomes all attached to the outside of the membrane. That's why it's called rough endoplasmic reticulum. Okay. What happens in the rough endoplasmic reticulum is these proteins are, they're made, and then they enter the endoplasmic reticulum for packaging. Okay, so for an example, streptomycin is a protein inhibitor and binds to a part of the bacterial rRNA. So this is an antibiotic. And it affects the ability for the bacterial ribosomes to carry out translation, to carry out protein synthesis. So if you can't carry out protein synthesis, you basically have a cell that cannot really function. So it inhibits protein synthesis and thereby inhibits the function of that bacterium. Okay, so the role of gene activation in protein synthesis. So we know the nucleus contains the chromosomes. When the nucleus, or when the cell is not dividing, those chromosomes are unwound. Okay, so you need to remember that they're only wound up into the chromosomal form during cell division process, cell replication. Okay, the chromosomes contain the DNA, and the DNA stores the genetic instructions for making those proteins. So that is essentially the job of DNA, is to code for protein. Okay, proteins determine the cell structure and function. So that is important to remember. The proteins determine the structure and function of the cell. Okay, so there's 3 major steps to protein synthesis. You definitely need to know these steps. We will spend time on these. Okay, transcription. This is when you copy the DNA instructions into mRNA or messenger RNA. So transcription, you are transcribing the language of DNA into the language of RNA. This happens inside the nucleus. And yes, you need to know these little details about where it happens. Translation, on the other hand, happens in the cytosol. So let's write that down, cytosol. And remember, cytosol is the fluid in the cell. And in translation, the ribosome, whether it's a free ribosome floating around in the cell or a fixed ribosome attached to rough endoplasmic reticulum, these ribosomes read that code on the mRNA and translate that into the language of proteins. And what helps in translation is another RNA molecule, tRNA. tRNA stands for transfer RNA, and it transfers individual amino acids to the growing polypeptide chain. And so I have a video that shows this, and it will really, I think, be helpful. Then you have the processing. And RER, rough endoplasmic reticulum, that's what that stands for. So there is processing that happens in the rough endoplasmic reticulum, and then it sends that protein that's been partially processed to the Golgi apparatus for further processing and packaging, and it's sent off to different areas of the cell or to outside of the cell. Okay, so starting with the first step, transcription. Transcription occurs where? Okay, you guys, do you remember? I know you can look at the last slide.
The nucleus.
In the nucleus. Excellent. Thank you. Okay. If I can get this going again. Okay, so the first step to transcription is we need to activate the gene we want to copy. So that gene, that DNA that houses that gene uncoils, removes the histones, because remember, it's coiled around those proteins that are called histones. So it uncoils, removes the histones, and you have a start and stop code for each gene, or a promoter would be the start code. And so those mark the location of the gene. And between the start and the stop code is the coding strand of the DNA that codes for that particular protein. You have a template strand that's used by the RNA polymerase. So there's a picture of RNA polymerase coming up. but the RNA polymerase plays an important role in copying the language of DNA into the language of mRNA. So transcription happens in the nucleus. And so what happens here is we are copying, so the DNA strands, look over here, basically unzips. They're kind of like a zipper. So if you unzip it, you have those nucleotide bases that are free on either side. And what happens is you are able to form an mRNA strand, a complementary strand based off of that open DNA. So the enzyme RNA polymerase is what is responsible for transcribing DNA. So you need to know what RNA polymerase is. You need to know that it is an enzyme and that it is what is responsible for transcribing the DNA into mRNA. So it binds to the promoter or the start sequence of the gene and then reads that DNA code for the gene. And as it's doing that, it binds nucleotides, complementary nucleotides, as you can see right here, to form the messenger RNA or the mRNA. mRNA duplicates that DNA coding strand, but remember, RNA does not have thymine. Instead of thymine, it's uracil. So on the RNA strand, the mRNA strand, thymine is replaced by uracil. So that conversion takes place right there in the nucleus. Then you have processing of the RNA molecule, the mRNA, and this is still happening inside the nucleus. So what happens is, even though you've copied this coding, this gene from the DNA, there are portions of that gene that do not code for anything. Those are called introns. So introns, come on, whoa, I don't know how I did that, sorry. I'm trying to draw and it's being silly. I don't know what's going on. Bear with me here. Okay. Introns, non-coding. The parts that are coding are the exons that code for protein. So you need to get rid of this junk DNA or these introns before you can actually go on to translation. Because if you code for those junk DNA, the introns, that gene is not going to make any sense. So you have to get rid of those portions that do not code for anything. So as we have reached the stop signal, the mRNA, the newly formed mRNA, detaches from the DNA molecule. And then you have the RNA processing. And this is where you get rid of those non-coding sequences, those introns. So the introns go out, okay? And you have the coding sequences, the exons, you splice them together into one complete gene or one complete mRNA that codes for the gene with no junk DNA in there. Now, what's important to know is that a sequence of 3 nucleotides is called a codon. So for example, if I just draw, see A, C, U, G, G, A. Okay, so this is just a small little sequence on the mRNA molecule. Each set of 3 is a codon. Each set of 3 is a codon, and it codes for 1 amino acid. okay? So we're going to talk more about this as we continue. But knowing that each triplet or set of 3 nucleotides in that mRNA is going to code for one specific amino acid. Okay, so this is kind of what it looks like in the transcription process. And this is in the DNA, this is in the nucleus. You have your RNA polymerase right here, finds the promoter sequence. Remember, the promoter sequence is a sequence of 3 nucleotides that basically says start here. And then it starts reading that sequence, and as it goes, it starts creating the newly formed mRNA molecule with the complementary strand. Complementary meaning that it forms complementary base pairs to the original DNA molecule that's being read. Once it has reached, the RNA polymerase has reached the stop codon, it releases, it dissociates, and it releases that newly formed mRNA strand. Again, important to remember that thymine is replaced by uracil in RNA molecules. And so after you've formed this mRNA strand, then we go back to splicing, processing, where we take out those introns that are junk DNA, splice together the exons, which is the actual coding sequence, and then we get our complete mRNA molecule. Once that is complete, that mRNA moves from the nucleus to a ribosome in the cytoplasm or attached to rough endoplasmic reticulum. So the mRNA relies on the ribosome for making protein, for the process of translation. Okay, so in the cytoplasm, there are just floating amino acids everywhere. Right, I have the cat and the dog in here. Okay, so the mRNA binds to the ribosomal subunits. You have a large subunit and a small subunit that come together, and it basically forms an assembly line for protein synthesis. And so down here, here's our mRNA. The big green blobs is the ribosome. And the mRNA kind of goes between the 2 ribosomal subunits, and the tRNA molecules, these guys right here, bring in individual amino acids. And on their base, on the bottom portion down here, they have a codon, which codes for one amino acid. So you have these tRNA molecules floating around in the cytoplasm. Each one is specific for a particular amino acid, and it has a specific anticodon, which is what it's called, that has to match just right with the mRNA molecule. If it matches with the codon on the mRNA, then you begin that process of protein synthesis or translation, where that tRNA molecule transfers that amino acid that it matches to the growing polypeptide chain. We have a video for this to watch, and you can see the process happening, and that I think it will be helpful for you. So remember, the tRNA molecule, the base right here is the anticodon, and the codon is on the mRNA molecule. So the anticodon has to match the codon with complementary base pairs. If it matches, it binds and then transfers the amino acid it's carrying to the growing polypeptide chain. Okay, with the help of enzymes, it joins the amino acids together with peptide bonds. And I know you guys know about peptide bonds. Those are important to continue to remember. Okay. Polypeptide chain has a very specific sequence of amino acids, just like the mRNA had a very specific sequence of nucleotides. If that sequence of amino acids is off, even by one amino acid, that can change the entire function of that protein. Okay, when you reach the stop codon, these components separate and you have formed your primary protein structure. So here is another illustration for translation showing we have a growing polypeptide chain. We have tRNA molecules that are floating around that have very specific anticodons. If the anticodon matches with the codon of the mRNA molecule, then that amino acid that has been carrying is transferred to the growing polypeptide chain. And then that just continues. Okay, so here is just a very simple illustration to remind you where these processes occur. Transcription happens in the nucleus. This is where you are transcribing DNA into mRNA. mRNA leaves the nuclear pores into the cytoplasm, where it meets up with ribosomes and carries out translation. And that is the process of protein synthesis. So knowing where these processes occur, what is happening at each step is very important. So at some point we'll have our video that will help make more sense of this. Okay, so how the nucleus controls cell structure and function. There is through direct control, through the synthesis of structural proteins and secretions. So in response to an environmental change, that would be direct control. Or it can control cell structure through indirect control over metabolism through enzymes. Okay. What do I need to know? What have we gone through so far? This is a great illustration. Again, I like this one too. You need to know what happens in transcription. Organelle structure and function, we'll talk more about those as soon as we finish with transcription and translation. Translation, what mRNA is and does and where it's made. tRNA, the same thing. What an intron is, what an exon is, what the splicing process, what happens there. Okay, so we're going to take a break and watch a video on transcription and translation. It's from my favorite YouTuber. Let's see if I can get it to work. Okay, 1 second. I need to share my screen with you. Okay, we have to listen to the ad for 5 seconds.
Manage your team's work easily with Monday.com. Just go to Monday.com and start tracking.
Okay, roll it. You know what this is? It is the longest word in the world. Like anywhere, any language, more than 189,000 letters. If you were to write it down, though I don't know why you would, it would fill up more than 100 pages. And if you could actually say it without like breaking your face, it would take about 5 hours. What the frick is this word? Well, it is the name of the longest known protein on Earth, and it's actually in you right now. Because of its enormous size, it was given the name Titan by scientists, and that's with 2 eyes. And it's a protein that helps give something like the springiness to your muscles. Today we're going to be talking about DNA and how it, along with 3 versions of its cousin RNA, unleash chemical kung fu to synthesize proteins just like this. So this is going to take a while to explain. So how about we make ourselves some Hot Pockets? One of my favorite. and cheese. Every time I take a bite, I wonder, how did they do it? How did they pack exactly the same flavor into every foil cardboard wrapped food-ish item? Clearly, there's got to be some super secret instruction manual kept in a location known only to 2 people. Since I'm talking about biology here, that brings up a related question. How did I get built from the DNA instructions and biological molecules we've been talking about? Today, that's what I'm going to do. Not actually make Hot Pockets or a person, but I'm going to be talking about DNA transcription and translation, which is how we get made into the delicious things that we are today, though hopefully none of us know how delicious people are. Animals, plants, and also Hot Pockets really are nothing more than salty water, carbohydrates, fats, and proteins combined in precise proportions following very explicit instructions. Let's say I want to make my own Hot Pocket. I would have to, 1, break into the lair of the Hot Pocket company holding the secret manual. 2, read the instructions on how to make the machinery to produce the Hot Pocket and the proportions of the ingredients. 3, quickly write down that information in shorthand before I get caught by the Hot Pocket police. 4, go home, follow the instructions, build the machinery, and mix the ingredients together until I have a perfect Hot Pocket. And that is how we get us. Very simply, inside the cell's nucleus, DNA instruction manual is copied gene by transcription onto a kind of RNA and then taken out of the layer where the instructions are followed by the process of translation to assemble amino acid strings into polypeptides or proteins that make up all kinds of stuff, from this titan down here to the keratin in my hair. But most polypeptides that get made aren't structural proteins like hair, they're enzymes which go on to act like the assembly machinery, breaking down and building and combining carbohydrates, lipids, and proteins that make up variations of cell material. So enzymes are just like whatever ingenious machinery they use in the factory to make this. Okay, let's start out in the layer, in the nucleus. The length of DNA that we're going to be transcribing onto an RNA molecule is called our transcription unit. And let's say, in today's example, that it's going to include the gene that transcribes for our friend Titan, which in humans at least occurs on chromosome 2. Now each transcription unit has a sequence just above it on the strand, and that's called upstream. Biologists call that upstream on the strand, and that sequence sort of defines when the transcription unit is going to begin. This special sequence is the promoter, and it almost always contains a sequence of 2 of the 4 nitrogenous bases that we talked about in our last episode: adenine, thymine, cytosine, and guanine. Specifically, the promoter is a really simple repetition. We got thymine, adenine, thymine, adenine, and then A, and then on the other side, A, because you know how this works, right? This is called the Ta-Ta box. It's nearly universal, and it helps our enzyme figure out where to bind to the strand. Now you'll remember from our episode about DNA structure that DNA strands run in one of 2 directions, depending on which end of the strand is free and which end has a phosphate bond. One direction is 5' to 3', and the other direction is 3' to 5'. In this case, upstream means toward the 3' end, and downstream means toward the 5' end. So the first enzyme in this process is RNA polymerase, and it copies the DNA sequence downstream of the Ta-Ta box. That's towards the 5' end, and copies it into a similar type of language, messenger RNA. Quick aside, so you'll notice that to read the DNA in order to make enzymes, we need an enzyme in the first place. So it kind of gets chicken egg here. We need the enzyme to make the DNA and the DNA to make the enzyme. So where did RNA polymerase come from in the first place if we haven't made it yet? What an excellent question. It turns out that all of these basic necessities get handed down from your mom. She packed quite a lot more into her egg than just her DNA. So, we had a healthy start. So thanks, Mom. So the RNA polymerase binds to the DNA at that Ta-Ta box and begins to unzip the double helix. Working along the DNA chain, the enzyme reads the nitrogenous bases, those are the letters, and helps the RNA version of those nitrogenous bases floating around in the nucleus to find their match. Now you might also recall from our previous episodes that nitrogenous bases only have one counterpart that they can bond with. But RNA, which is the pink one here, doesn't have thymine like DNA does, which is the green and the blue. Instead, it has uracil. So U appears here in T's place as a partner to adenine. As it moves, the RNA polymerase rezips the DNA behind it and lets our new strand of messenger RNA peel away. Eventually, the RNA polymerase reaches another sequence downstream called a termination signal that triggers it to pull off. Now, some finishing touches before this info can safely leave the layer. First, a special type of guanine is added to the 5' end. That's the first part of the mRNA that we copied, and that's called the 5' cap. On the other end, it looks like I fell asleep with my finger on the A key of my keyboard, but another enzyme added about 250 adenines onto the 3' end. This is called our poly A tail. These caps on either end of the mRNA package make it easier for the mRNA to leave the nucleus. They also help protect it from degradations from nearby passing enzymes, while also making it easier to connect with other organelles later on. But that's still not the end of it. As if to try to confuse me to protect the secret Hot Pocket recipe, the original recipe book also contains lots of extra misleading information. So just before leaving the nucleus, that extra information gets cut out of the RNA in a process called RNA splicing. And it's something like editing a video. This process is really complicated, but I just had to tell you about two of the key players because they have such cool names. One, the SNRPs, which are small nuclear ribonucleoproteins. These are a combination of RNA and proteins, and they recognize the sequences that signal the start and end of the areas to be spliced. SNRPs bunch together with a bunch of other proteins to form the spliceosome, which is what does the actual editing, as it were, breaking the junk segments down so their nitrogenous bases can be reused in DNA or RNA, sticking together the two ends of the good stuff. That good stuff that gets spliced together, by the way, are called the exons because they will eventually be expressed. The junk that gets cut out are just the intervening segments or the introns. The material in the introns will stay in the nucleus and get recycled. So, for instance, Titan down there is thought to have hundreds of exons when it's all said and done, probably more than 360, which may be more than any other protein. And it also contains the longest intron in humans, some 17,000 base pairs long. Man, Titan, it is just a world record holder. So now that it's been protected and refined, the messenger RNA can now move out of the nucleus. Okay, so a quick review of our Hot Pocket Mission Impossible caper so far. We've broken into the layer containing the instructions, we copied down those instructions in shorthand, we added some protective coatings, and then we cut out some extra notes that we didn't need, and then we escaped back out of the layer. Now I have to actually read the notes, make machinery, and assemble the ingredients. This process is called translation. So next, rewind your memory or just watch that video again to the episode about animal cells. Do you remember the rough endoplasmic reticulum? I hope you do. Those little dots on the membranes are the ribosomes. In the process, messenger RNA gets fed into a ribosome like a dollar bill into a vending machine. Ribosomes are a mixture of protein and a second kind of RNA called ribosomal RNA or rRNA. And they act together as a sort of workspace. rRNA doesn't contribute any genetic information to the process. Instead, it has binding sites that allow the incoming mRNA to interact with another special type of RNA, the third in this caper called transfer RNA or tRNA. And tRNA really might as well be called translation RNA because that's what it does. It translates from the language of nucleotides into the language of amino acids and proteins. On one end of the tRNA is an amino acid. On the other end is a specific sequence of nitrogenous bases. These two ends are kind of matched to each other. Each of the 20 amino acids that we have in our body has its own sequence at the end. So if the tRNA has the amino acid methionine on one end, for instance, it can have UAC as the nucleotide sequence on the other end. Now it's just like building a puzzle. The mRNA slides through the ribosome reads the mRNA 3 letters at a time, each set called a triplet codon. The ribosome then finds the matching piece of the puzzle, a tRNA with 3 bases that will pair with the codon sequence. That end of the tRNA, by the way, called the anticodon. Sorry for all the terminology. You need to know it. And of course, by bringing in the matching tRNA, the ribosome is also bringing in whatever amino acid is on that tRNA. Okay, so starting at the 5' end of the mRNA that's fed into the ribosome, after the 5' cap, for almost every gene, you find the nucleotide sequence AUG on the mRNA. The ribosome finds a tRNA with the anticodon UAC, and on the other end of that tRNA is methionine. The mRNA, like a mile-long dollar bill, keeps sliding into the ribosome so that the next codon can be read, and another tRNA molecule with bright anticodon binds on. If the codon is UUA, then the matching tRNA has an AAU on one end and a leucine on the other. And if the mRNA has an ADA, then the matching tRNA has a UCU on one end and an arginine on the other. In each case, that new amino acid gets connected on the previous amino acid, starting a polypeptide chain, which is the beginning, very beginning of a protein. But it turns out that there are lots of different ways to read this code, because UUA is not the only triplet that codes for leucine. UUG does too, and arginine is coded for by 6 different triplets. This is actually a good thing. It means we can make a few errors in copying and transcribing and translating DNA, and we won't necessarily change the end product. The process continues with the mRNA sliding in a bit more, and the ribosome bringing in another tRNA with another amino acid, that amino acid binding to the existing chain, and on and on and on and on and on. Sometimes for thousands of amino acids to make a single polypeptide chain. For example, this whole word is basically just the names of the amino acids in the sequence and the order in which they occur in the protein, all 34,350 of them. But before we can make our own Hot Pockets, that string of amino acids becomes my muscle tissue, we have some folding to do. That's because proteins, in addition to being hella big, can also contort into very complex and downright lovely formations. One key to understanding how a protein works is to understand how it folds. Scientists have been working for decades on computer programs to try and figure out protein folding. Now, the actual sequence of amino acids in a polypeptide, what you see scrolling along down there, is called its primary structure. One amino acid covalently bonded to another, and that one to another, and to another in a single file. Some amino acids don't like to just hold hands with two others. They're a bit more promiscuous than that. The hydrogens on the main backbone of the amino acids like to sometimes form bonds on the side, hydrogen bonds, to the oxygens on amino acids a few doors down. When they do that, depending on the primary structure, they bend and fold and twist into a chain of spirals called helix. We sometimes also find several kink strands laying parallel to one another called pleated sheets. All those hydrogen bonds and pleated sheets are what makes silk strong, for instance. So in the end, our promiscuous amino acids lead to wrinkled sheets. These hydrogen bonds are what help give these polypeptides their secondary structure. But it doesn't end there. Remember the R groups that define each amino acid? Well, some of them are hydrophobic. Since the protein is in the cell, which is mostly water, all of those hydrophobic groups try to hide from the water by huddling together. And that can bend up the chain some more. Other R groups are hydrophilic, which, if nothing else, means they like to form hydrogen bonds with other hydrophilic R groups. So we get more bonding and more bending, and our single file line has now taken on a massively complex three-dimensional shape. It also explains why I can fix my bed head by wetting my hair with water. Water helps break some of those hydrogen bonds in keratin, which relaxes its structure. That way I can comb it out, and when it dries, those bonds reform, and voila, perfect hair. All of this shape caused by bonding between R groups gives our polypeptide its tertiary structure. So now we have a massively contorted polypeptide chain, and it actually contorts very precisely. Sometimes just one chain is what makes up the whole enzyme or protein. In other proteins, like hemoglobin, several different chains come together to form a quaternary structure. So quick review of structure. Sequence is the primary structure, the backbone of hydrogen bonds forming sheets and spirals. Secondary structure, R group bonds are tertiary in the arrangement of multiple proteins together, the quaternary structure. These polypeptides are either structural proteins, like this thing at the bottom here, you can find in your muscle or in my hot pocket. They might also be enzymes. Enzymes like do stuff. They can cut up biological molecules like I do with the chef's knife. They can mix stuff. They can put stuff together. So from that one recipe book, you get all the ingredients and all the tools necessary to make me, which is better than a hot pocket. Would you all agree? Now take your time with this stuff. Feel free to watch the episode a couple of times, because next week we're going to talk about how cells swap all of this genetic information through reproduction. Thank you for watching this episode. Bye now.
Okay. Oh, let's go back to where we were. Ooh. Any questions about that video? Okay, let's take a break, and I'm going to take roll. So when I call your name, please. Oh, there is a question. I said, oh, you said we were ahead of schedule. Did you say major cell structures be on the first test as well as everything else we're coming? No. So, oh, I don't have my syllabus right here with me. Let's go check that together. I'll just check the schedule real quick. If I can scroll up. There we go. Physiology schedule. Okay, so just as a reminder, your quiz is due today by midnight over macromolecules. Your first exam your first exam is going to include the cell, but not cell transport, because we definitely, well, Actually, honestly, I already wrote the exam. So the cell will be on the second exam, if that helps. So the first exam has already been written and it will not have the cell in it. It will be on the next exam. Let's see, here's another question. Okay. Let's go back to our slide. Okay. So when I call your name, please unmute so that I can mark you as being here, okay? Kayla.
Here.
Thank you. Reagan. Reagan Casserly.
Here.
Oh, yep. I saw you were the one who wrote a note. Okay. Kelsey Amal. Is Kelsey here? Okay. Tana.
I'm here.
Thank you. Bailey. Is Bailey Fulmer here? Okay. Stevie.
I'm here.
Thank you. Josh.
I'm here.
Thank you. Joel. Joel Langemeyer, I see you.
Here.
Okay, I see
Yeah,
that you're
here.
here. Okay.
Sorry, you cut out.
Oh, okay, sorry. Taylor Lobeda.
I'm here.
Thank you. Taylor Lavelle.
Here.
Thank you. Grace.
Here.
Thank you. Andrew Marr.
Here.
Thank you. Amber Michael. Amber. Okay. Jimmy Nguyen. Nothing, honey. Okay. Yuen Nguyen.
I'm here.
Thank you. Marissa Pacha.
Here.
Thank you. Jamie Plague. Plague. Is Jamie here? Okay. And Jeanette.
Here.
Thank you. Morgan.
Here.
Thank you. Macy.
Here.
Ayla.
Here.
Elijah.
Here.
Araya.
Here.
Ava. Awesome. All right. Thank you guys. So we got a request to go back to how the nucleus controls cell structure and function. So let's see if we can go back to that real quick. If I can get my screen. How organelles, how nucleus controls, oh, is that right here? Okay. So how nucleus controls cell structure and function. So it's either direct control or indirect control. So direct control is through the synthesis of structural proteins and secretions. So they can secrete substances as a response to the environment. Indirect control is through over-metabolism through the use of enzymes. Are there questions on this? We just wanted to look at this slide again. Okay. So that is our summary slide of transcription and translation. We are going to start moving into other types of organelles that you find in the cell. So 5 types of membranous organelles. So these are organelles that are bound by a membrane. So endoplasmic reticulum, and that can be either smooth endoplasmic reticulum or rough endoplasmic reticulum. The Golgi apparatus, lysosomes, peroxisomes, and mitochondria. So we are going to talk about each of these separately, and you will need to know these organelles, their functions, and yeah, basically their functions, because you have that, remember this picture of the cell at the beginning of this lecture? You will have one picture to identify structures that we talked about at the beginning of this lecture. I am recording this lecture and I will upload it to YouTube and send the link on Canvas like I've been doing, so you can always go back and re-watch it to kind of refresh your memory if you need to. Okay, so starting with the rough endoplasmic reticulum, this is covered by ribosomes. It gives it the actual rough appearance when you're looking at it under a microscope. It is active in protein synthesis because ribosomes are directly involved in protein synthesis. You can't have protein synthesis without ribosomes. And at the rough endoplasmic reticulum, you have that polypeptide chain that forms, and then it folds that protein into secondary and tertiary structures. It doesn't fold it all the way into a quaternary structure, just secondary and tertiary structure. So it does some folding in the rough endoplasmic reticulum. It then encloses those proteins that are partly formed into transport vesicles that then travel to the Golgi apparatus. And the Golgi apparatus we'll talk about in just a second. The smooth endoplasmic reticulum, on the other hand, does not have ribosomes. And that's why it's called smooth endoplasmic reticulum, because it actually looks, it actually looks smooth. So no attached ribosomes, and it is responsible for synthesizing lipids, steroids, and hormones. So completely different function. Also important in detoxifying drugs and alcohol. So quick question, if the smooth endoplasmic reticulum, whoops, hold on. Oh shoot, I'm sorry. I need to go back to screen sharing. Where was I? I'm sorry, I lost our spot. There we are. I was just trying to find our chat. So if smooth endoplasmic reticulum is involved in detoxifying drugs and alcohol, what major organ do you think you would find it in? A lot of it.
Liver.
Liver. You're right. You would find a lot in the liver. Very good. Now if I can get this, there we go. Okay, so detoxifies drugs and alcohol. There's an enzyme, P450, that helps in some of the drug detoxification. In muscle fibers, the smooth endoplasmic reticulum is called the sarcoplasmic reticulum, and it stores calcium. And so when we talk about muscle physiology, we'll be talking about the sarcoplasmic reticulum and the role of calcium in muscle contraction, but we are not there yet. Okay, so here is an illustration showing you the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. So they look different, they have different functions. Here are actual electron micrograph pictures showing the rough endoplasmic reticulum and the smooth endoplasmic reticulum. You can see how very different they look. Okay, the Golgi apparatus. So we'll talk a little bit about the Golgi apparatus, and then we have a short video clip to watch to help illustrate what's going on with the Golgi apparatus, because it can be a little confusing as well. So functions, it modifies and packages secretions. So it receives those partially formed proteins from the rough endoplasmic reticulum, for example, and it modifies them even further and then packages them to go to different parts of the cell or to go to outside of the cell. So secretions like proteins, hormones, or enzymes for the release from the cell. Can package special enzymes within vesicles, so forming lysosomes, for using in the cytoplasm. So how this works, the Golgi apparatus has two sides. You have the forming phase, or the cis phase, and the maturing phase, or the trans phase. You need to know the difference between the cis forming phase and the trans maturing phase. I will ask you a question about that. Secretory vesicles, they modify and package products for exocytosis. You have membrane renewal vesicles, which basically add or remove components of the plasma membrane. You have lysosomes, which carry enzymes to the cytosol, which is the fluid inside the cell. So here we have this wonderful picture. It is a really great picture, by the way. It illustrates what's going on really well. So you have transport vesicles that are coming from, let's say, the rough endoplasmic reticulum with a partially formed protein. It joins up with the forming phase. The forming phase is the cis phase, so we just want to remember that. So cis, and this is the trans phase here. And then these products travel through the Golgi apparatus where they're modified and then packaged and then sent out to different parts of the cell or to the outside of the cell. So the three main ones, we have made lysosomes, secretory vesicles for releasing products outside of the cell, and then membrane renewal vesicles, which add or remove components to the plasma membrane itself. Okay, so let's watch this little clip. I think it's like 3 minutes long. And I think it will help. I have to share this, sorry. 1 second. Okay.
It is important that translated proteins are delivered to their specific cellular location. To accomplish this, the protein is transferred through a series of membrane structures. A principal membrane component is the Golgi apparatus. The Golgi apparatus is the sorting organelle of the cell. Proteins from the rough endoplasmic reticulum are sent to the Golgi. As the proteins move through the Golgi apparatus, they are modified and packaged into vesicles. Because the Golgi apparatus receives proteins from one location and targets them for delivery to a second location, it is sometimes considered the post office of the cell. The Golgi apparatus consists of general components. The cis cisterna nearest the endoplasmic reticulum, or ER, the medial and trans cisternae, and the trans-Golgi network. Other key players in this process are the proteins being transported and the enzymes that modify them. Translated proteins are encapsulated in vesicles in the ER. A group of these vesicles fuse, and these fused vesicles form the cis cisterna. As the protein moves through the stack, it is modified by resident Golgi enzymes at specific locations in the apparatus. These modifications are important because they provide the signal that determines the final destination of the protein. So how does the protein move through the Golgi? Movement occurs in waves. First, the cis cisterna becomes part of the medial Golgi cisternae. Behind it, a new cis cisterna is formed by the fusion of vesicles from the ER. Meanwhile, one of the medial cisternae migrates and becomes the new trans cisterna. Collectively, this process is known as the cis maturation model. Proteins are sorted within the trans Golgi network. Proteins with the same target sequence are destined for delivery to the same location. The trans Golgi network then buds off into vesicles. These vesicles then migrate to their target location. These locations include internal organelles, such as the lysosome, the digestive organelle of the cell. The vesicles can also be targeted to the cell membrane, where the targeted protein can be released from the cell for delivery elsewhere in the organism.
Okay. Short and sweet. I like that video. So hopefully that helped kind of illustrate how the Golgi apparatus functions. Its role in processing and sending out information and proteins, forming lysosomes, secretory vesicles, and membrane renewal vesicles. So a very important function. So you will need to know how the Golgi apparatus functions, the cis phase, the trans phase, and what's happening there. Okay, other organelles in the cytoplasm. Lysosomes, which we just learned are formed by the Golgi apparatus. And these have enzymes in them. And they, as she said in the video, they're like the digestive organs of the cell. They break down old products. They break down things that don't belong in there or have died or are no longer useful. So they can also destroy things like bacteria, break down molecules, and recycle damaged organelles. Okay. Peroxisomes. These are enzyme-containing vesicles, and they also function to break down substances, but just different substances. So they can break down fatty acids and organic compounds. I would never ask you to identify a peroxisome or a lysosome because they're just kind of bubbles within a cell. They're not very specific looking. Peroxisomes produce hydrogen peroxide, and that's what they use to break down these organic compounds and fatty acids. They replicate by division, so they're basically just splitting in half, and now you have 2 peroxisomes. Okay, another important organelle in the cell is the mitochondria. And the mitochondria has its 2 layers of membranes, an inner membrane and an outer membrane. The inner membrane is heavily folded. These folds are called cristae. And within the cristae, within the inner folding, you have what's called the matrix. It's the fluid in the within the second heavily folded membrane. What happens at the mitochondria is that it takes the chemical energy from food, glucose specifically, breaks that down through a series of chemical reactions and releases or makes ATP, adenosine triphosphate, the energy molecule for our bodies. So mitochondria are incredibly important and for our function and our ability to do anything, to move your muscle, to stay awake, to talk. All of that requires energy in the form of ATP, and you get that ATP from the mitochondria. So we will talk about cellular respiration in a couple chapters, and this is specifically about how ATP is made in the mitochondria. So we will spend more time on this later. Okay, in mitochondria it functions with aerobic metabolism. Aerobic meaning it's using oxygen. So mitochondria use oxygen to break down food and make ATP. This is the general equation for aerobic metabolism. This is as much as you will need to know for this chapter as far as metabolism is concerned, but you do need to know this general equation. Glucose, C6H12O6 plus oxygen, you go through a series of reactions, releases carbon dioxide as a waste product, water as a waste product, and ATP as your energy molecule. There are 3 stages to aerobic metabolism. The first is glycolysis, where you're converting glucose to pyruvic acid. The second is the Krebs cycle. Usually we just refer to it as the Krebs cycle, not the tricarboxylic acid cycle. And this is where you convert pyruvic acid into carbon dioxide. And then you have the electron transport chain, which happens on the inner membrane of the mitochondria, and that is where the majority of the ATP is made. So again, we will talk about this in detail later. Other organelles in the cytoplasm. Oh, one last thing to say about mitochondria. Produces 95% of the ATP the cell needs to stay alive. That's a huge amount. So mitochondria have a very important role. They're often referred to as the powerhouse of the cell because they're responsible for producing that much energy. Moving to centrioles, these are organelles within the cell that form what's called the spindle apparatus during cellular division. So in this little picture here, you can see these lines. That's forming the mitotic spindle. These fibers are attached to the centrioles on either side. So it has an important role in cell division. Otherwise, it's not doing much in the cell. Okay. We are going to talk about the phospholipid bilayer, the plasma membrane. I'm kind of thinking this might be a good place to stop. We have just a couple minutes left for today. Let's stop with this and start with this again on Thursday. But for now, are there any questions? You can chat or you can say them out loud. Are you doing your schoolwork? I'm telling you, she's just reading books. You need to text your teacher. I will. Okay. Tell him you don't have your charger. Sorry, excuse me. My son is home today. He had to do another COVID test Saturday because he had a fever on Friday. So he is doing homeschool today while I am teaching. And so I apologize for that. Okay, I am not getting any questions from anybody. Do we feel confident about transcription and translation? About the Golgi apparatus? Any questions about the quiz or exam?
Is there a place that has what pages we're supposed to read out of the textbook?
It doesn't have specific pages, it has chapters, if that helps. If you hold on, I'll go grab my textbook and look for you right now, okay? So are you talking specifically about this chapter?
Yeah, that and the, I know with the last one I had to look through the textbook to try to find the pages that had no, I can't remember what we just did for the section.
That's okay. Basically,
Molecules.
molecules. Okay, the macromolecules.
I was trying to look through the textbook to try to find what pages to read. I just didn't know if there was a place that had basically what specific pages we needed to read.
No, there's no specific
just-
page numbers that are written down anywhere, but what we are talking about right now is in chapter 3.
Okay.
So the beginning of chapter 3 is what we're talking about now, and then our next lecture, when we finish this one, will be on diffusion and osmosis, which is still chapter 3. So
Okay.
there's quite a bit of chapter 3 we're doing right now. The macromolecules was the end of chapter 2.
Okay.
Does that
Yeah,
help?
I think I found that one. Yeah, I
Okay.
think I found that one. I just was looking around for a while the other night trying to find a spot that had like what specific pages. Like I know you have a thing that says, I think on like the syllabus that says read certain chapters, and I don't know. I was just trying to find if there was a spot that
Page
had
numbers.
page numbers and whatnot.
No,
but
unfortunately,
Okay.
no page numbers. But if
Okay.
you're ever stuck and you can't find something, you can always text me and I'll help you out.
All right, sounds great.
Okay. Anybody else? No questions about the quiz that's due today? Please make sure you take your quiz today. Don't wait till the last minute, and make sure you study beforehand. Please. I want you to have the best chance possible to do well. There are 21 questions. Well, 20 questions, 21 points. And it's covering macromolecules. So I would study that chapter very closely, and you should be able to do okay, I think.
Is that all it's over, is the macromolecules?
Yes. Because I accidentally wrote your test first and did everything, and then I realized, wait, you don't have a test this week, it's a quiz, and I went back and rewrote the quiz. So it should be just the macromolecule section, that chapter. If that makes sense. Okay. So it's the last half of chapter 2 in your textbook. Okay. All right, guys, if that is it, I'm gonna let you go. It's right about time to go anyways. I will see you on Thursday. I do not know if it will be Zoom or in class lectures, so please just pay attention to your announcements. I will make sure to let you know as soon as I know, because right now we're supposed to be self-isolating because of my son. So until we have those results, we're still Zooming. Thank you for being so flexible. I really, really appreciate it. And have a fantastic day. Good luck on your quiz. Bye, guys.